we're covering the electron transport chain / OxPhos pathway. And I do expect you to be able to draw and go over all of the various parts. And so rather than draw out the membrane, and which membrane does this occur in? What membrane of the cell? The inner membrane of mitochondria. Okay. And we have complex 1, kind of looks like Florida. Or the mirror image, at least, the way I have it drawn here. And what's the initial electron donor in complex 1? Like, what's down here? NADH. So it's NADH. And technically, with a proton, that goes on to become NAD+. Okay? And then there are a series of iron-sulfur proteins. There's several of them that go and transfer the electrons along complex 1. And then there's a protein, specific iron-sulfur protein, that's up there right in the portion of it that looks more like the panhandle of Florida. And what's that one called? What is that important protein of this multi-subunit protein complex? Pardon? Nope. No, ubiquinone and ubiquinol are not proteins. Those are that's coenzyme Q. It's what? No. It's got a single letter and number name. Kind of sounds like something from, I don't know. No. Good guess, though. It's the N2. Yeah, right. It's N2.
My second
Uh-huh. It rhymes with C1? Okay, I can see someone who needs to retake some English classes. All right, but this is what's going to pass off the FMN was along with the FES iron proteins, but this is what passes it off to the coenzyme Q. Which remember, coenzyme Q is actually inside the membrane already because it doesn't leave. So we have CoQ becoming ubiquid. If we're going from ubiquinone to ubiquinol, which is CoQH2. Okay. And where does the CoQ go from now on? Not for now, next. Complex 3. So it's going to go, on to complex 3. Because I don't have a big enough thing to write. And once again, we're still in the membrane. The little balloon-looking things. We have complex 3.
So those can go right next to it. Complex 1.
Right. As long as you number it, label it. Because I'm wanting to point out the part that is specific for the electrons from NADH. Okay, since there is a difference between the NADH and the FADH2. So this is complex 3, which is also called the BC1 complex or cytochrome BC1, and there's lots of parts to it, including the risky protein, which kind of looks like the little hammer. I know you can be jealous of my artistry. But the CoQ, and I'm not even about to try to draw the Q cycle. I do expect you to be able to draw it, but that would be like an inset. I definitely do not have enough room on this right here to draw it, but the Q cycle is going to occur down here. And so we have the CoQH2 coming in, and ultimately it's going to be leaving as a CoQ. But it's because, remember, one electron goes up through the risky protein to cytochrome BC1, and then ultimately to cytochrome C, which the cytochrome C is the one that's special because it's here, and it's the only one that's actually in the, I can't draw here, let me, nope, come on, maybe you can see it. It's the only one that's in the intermembrane space. But one electron goes up through the risky protein to the cytochrome C1, and ultimately passed on to cytochrome C, which is a separate protein from complex 3. The other electron goes through the heme Bl, heme BH, down to the N side of the complex 3, which is where you either already have a coenzyme Q waiting or a semiquinone. If it's halfway through the cycle, it's either going to be this or, the semiquinone. And so if it's a semiquinone, then that one electron makes it co-QH2, which it can then go on to another BC1 complex. If it's the quinone, it makes it to the semiquinone state where it can wait for the next electron. Okay. Once again, there's more detail to this than what I'm showing right there. But there's also another way to get electrons to the BC1 complex or complex 3, and that's using complex 2. Okay. So once again, I have to... So we have our membrane. And this complex is special in a couple ways. What's one way that's special? What's the initial donor of electrons here? Right. This is where FADH2 comes into play. And so we have FADH2 becoming FAD. Okay. And what is it passing the electrons off to? What's the recipient? It's coenzyme Q. And so we have here, once again, co-Q becoming co-QH2, which can then go on to the BC1 complex or complex 3. But now there's a fail-safe or stopgap there. What's another special thing about complex 2? There's the heme B, which I'm going to draw right here. So I'll just put a B there. And that's the one that's going to catch or be like the sponge in case any electrons are errant. It happens so quickly that sometimes they can get away from things, and it's there to where it can catch it in order to pass it off to, the waiting co-Q. All right. And if we go back here, what is missing in this instance? What else does complex 1 do? That's really important if you're going to make ATP. The protons move. Okay, and so we have protons that are being pumped, and it's an excess of protons, but the protons are being pumped to the outside. Okay, so make sure we want to draw that in. What about in complex 3? Are protons being pumped in? Pumped to the inner membrane space? Yes. So we also have protons going through to the inner membrane space, which, there we go. And finally, what about complex 2? Are there any protons being pumped across the inner membrane to the inner membrane space of complex 2? No. That's one other thing that makes it different. Okay, and so that's really how we got caught up so far with where we are right now. And so we can go back here, and of course this is once again the depiction that the book has for the Q cycle, and we went over that in greater detail in the last class. But let's continue on. So now we're up to complex 4. Okay, in complex 4 is cytochrome C oxidase, because ultimately we're going to be oxidizing cytochrome C. So cytochrome C literally jumps. It's in the inner membrane space, and it travels from the complex 3 to complex 4. So from the cytochrome C reductase, essentially to become cytochrome C oxidase. And the last postdoc lab that I worked at, where I worked on complex 3, we also had people in the lab that was working on complex 4. Because like I said, there are many diseases and disease states and pathologies that are attributed to mitochondrial defects. Okay. All right, and so we see here now we've got cytochrome C, which they're showing as this little dashed line. So it's jumped from complex 3 to complex 4, where it binds and it transfers the electrons to coppers. So there's a copper A, and it's instead of an iron sulfur center, it's an iron copper center using hemes, and these are called heme A and A3. I do not know why there's an A and an A3 and not an A2. Okay, that's the way it is. And then it goes to copper B. But yeah, this is different than what we've seen in the past where we had the FMN, the iron sulfur. This one's actually using copper, okay, instead of an iron sulfur center. You can see that we have oxygen, which why is it that oxygen is permeable for the membrane? We don't have to have an oxygen transporter to go across membranes. But small. When you say neutral, what do you mean?
Has no charge.
Well, that's not it. Because, it's nonpolar. Okay, so it's small, nonpolar molecule, so it can diffuse right across. And so then it comes... Whoops, I just did the wrong one. Sorry about that. It can come in and it accepts the electrons from the copper B. It also helps take out some more of the protons from the matrix side in order to make water. And at the same time, it pumps additional protons out. Okay, so remember, complexes 1, 3, and 4 move protons from the N side inside the matrix to the P side, or the intermembrane space. But complex 2 does not. All right, so then we've actually already discussed this part. The mechanism by which the proton gradient works is because the protons have to flow back into the matrix through the ATP synthase, which really looks like a turnstile. And the part that, to me, looks more like a ratchet, it's called the stator, but it kind of looks like a little ratchet, or it also reminds me of like the needle on an old turntable. Okay, and that's what helps with the movement as well as literally the axle of it. And it's this flow of the protons that forces the ADP and inorganic phosphate to form ATP. Okay. All right. And this is just a little cartoon depicting everything that we've said thus far in case you needed to see it in another format. So this is a structural summary of it, which I think is really cool that they've been able to put this all together. And the idea that, once again, if you're starting with... Now, the one thing I want to point out is if you're starting with NADH, you make coenzyme Q. There's a putative Q cycle that's happening here because they're really not for sure. They haven't been able to deduce exactly what's going on here. But please note, coenzyme Q cannot leave the membrane. They're just trying to show that it's going around, bypassing, complex 2. Okay. To complex 3, where the Q cycle occurs. At the same time, if you're starting with FADH2, then it's going to go through the citric acid cycle here, and ultimately to coenzyme Q to go to complex 3. Complex 3, once again, pumps out the protons. The ultimate recipient of those electrons, or what is reduced, is cytochrome C. Try not to get cytochrome C and cytochrome C1 mixed up. Cytochrome C1 is actually right around there. And cytochrome C is in the intramembrane space, and it goes to the cytochrome C oxidase, which is complex 4, which is where the coppers will take the electrons and will reduce oxygen to water. And once again, protons get pumped across the membrane.
You said we don't have to know how many.
I'm not going to ask you about the stoichiometry because different books will say different things. Like, is it really 4? Is it really just a net gain of 2? Or right? It is an excess. It's not a 1-to-1. Okay, so the way that shows makes it look like everything's really spaced out, but in reality, if you think back to last semester, remember we had those rafts and islands to where you can have proteins that kind of come together and to form like larger zomes. They always like to give it a zome ending. And so that also happens here to where they call them the respirasomes, and parts of the electron transport chain seem to form together, which makes sense, because that way they can pass the electrons off really quickly. Okay, and so, but it's large enough that you can see it microscopically. I will say it right. Okay, this comes to our shuttle mechanisms, because the NADH, which is from glycolysis, is in the cytosol, and the NADH or FADH2 from the mitochondrial matrix pathways are going to be in the mitochondrial matrix, and yet the electron transport chain is in the inner membrane it's in the inner membrane for the most part, except for cytochrome C. So we have to have ways to get the reducing reagents from the cytosol into the inner membrane. NADH, or FADH2 for that matter, cannot pass through the membrane directly. So we have to have ways around that. And so there are a couple of ingenious ways around that, one of which we've actually seen before, and we just may not realized it. Okay? So these shuttle mechanisms are going to transport metabolites. And we've already seen this, for example, with gluconeogenesis. You know, how oxaloacetate can't go directly through, and so we use malate, right? Convert it to malate, and malate can pass through malate and aspartate, but oxaloacetate cannot. Okay. First up is called the glycerol phosphate shuttle. And so once again, there's cytosolic NADH because of glycolysis. But we cannot cross into that holy of holies of the mitochondrial matrix. And so since it can't, there are two components that can. It's glycerol and our good friend DHAP, or I should say glycerol phosphate and our good friend DHAP, dihydroxyacetone phosphate. I would expect you to be able to draw these two, because glycerol, you should know, what does it look like? 3 carbons, and each carbon has an alcohol. And if it's a phosphate, one of those terminal carbons has been transformed into a phosphate. I mean, that's what it is. And DHAP, you already should know, because you draw acetone, which is 3 carbons, but it's a ketone in the middle. And then you have dihydroxy, means each of the other carbons has an alcohol, and once again, one of those alcohols has been turned into a phosphate. The glycerol phosphate shuttle only produces 1.5 ATPs for every NADH. But if you remember, we've already been saying along the lines that you can get up to 2.5 ATPs for every NADH. So what do you suppose is happening here? Why is it that we get short-shrifted, so to speak? What else must be being used here? Where have we seen the 1.5 ATPs before? It's FADH2. So what this means is the NADH electrons from the cytosol ultimately get passed to an FADH2, or to form FADH2 inside the mitochondrial matrix. Okay. All right. Originally, it was found in insect muscles, but our muscles can do it as well as our brain. Our brain uses glycerol phosphate shuttle. Okay, so this is what it would look like. We have our mitochondria there. We have glycolysis, happening here, producing the NADH. It also produces DHAP. I mean, that's, right there in steps 4 and 5. Okay. And so we have DHAP being transformed into glycerol phosphate. If you notice right here, the only difference is we go from a ketone to an alcohol. So, and it utilizes an NADH. So what class or classes of enzymes could this be? Dehydrogenase. And if you're going to name this, what would you name it? There's at least 2 possible names that you could think of. DHAP dehydrogenase or glycerol phosphate dehydrogenase. And if you notice, both of these components can go back and forth. And so, whoops. We see here that it's the glycerol phosphate dehydrogenase. And there are two different isoforms. The one in the cytosol uses NADH, but the one inside the mitochondrial matrix uses FAD. Okay? And so that's the reason why you only get 1.5 ATPs, because this FAD has to start with complex, FADH2 starts with complex 2. So you lose the ability of those extra protons being pumped through in complex 1. But the glycerol phosphate shuttle utilizes, you only get 1.5 ATPs from every NADH. However, I have already mentioned the fact that there must be some way to get up to 2.5 ATPs for every NADH, so there's another shuttle as well. That's our good friend, whoops, went too far, malate and aspartate. So we've actually already seen this. This is part of what metabolic pathway? The citric acid cycle. Okay, what else can it be used for? Maybe for glycolysis. That's not really the main use for malate or aspartate. So it's really citric acid cycle and gluconeogenesis, right? Because the malate becomes oxaloacetate. Oxaloacetate ultimately gets transformed to glucose. Okay, so this is found in kidney, liver, heart. And it's because they have low concentrations of glycerol phosphate dehydrogenase relative to some of the other tissues. Malate can cross the mitochondrial membrane. OAA, oxaloacetate, cannot. That's the reason why we see with gluconeogenesis the fact that it gets transformed to malate first. And aspartate can. If you use this shuttle, you get 2.5, because that means that both sides obviously use NADH. Okay. Hope you like the little school buses. Get it for the shuttles. I was trying to be cute. All right. So this one looks a little bit more complicated, but it's really not. We've actually seen all this before, so I don't know where we want to start. I'm going to start here with, well, we'll start with glycolysis. Glycolysis produced the NADH. We've got to get, well, it's frozen. There we go. We've got to essentially get the electrons from this NADH to the inside. So we have oxaloacetate becoming malate. And so what class or classes of enzymes could this be? Dehydrogenase. And what's the name of this one? That's malate dehydrogenase. Technically, it would be cytosolic malate dehydrogenase. Okay, then the malate gets... Okay, whoa, went too far. The malate gets transported across using the malate transporter inside the cytosol inside the matrix, to where a waiting NAD becomes NADH. Okay, which that can then go on into complex 1 and ultimately form 2.5 ATPs. And so this enzyme would be, since it turns a malate into oxaloacetate, mitochondrial malate dehydrogenase. Okay, so we made the oxaloacetate. Oxaloacetate becomes aspartate, and you can see what they look like here, in case you've forgotten, at the expense of a glutamate becoming an alpha-ketoglutarate. So what class of enzymes could this be? Oxaloacetate becoming aspartate. Aminotransferase. What specific aminotransferase is this one? This is aspartate aminotransferase. Technically, it would be the mitochondrial one. And then aspartate can go on out to where it can be transformed once again into oxaloacetate. And we can get the alpha-ketoglutarate to glutamate, and it's just the cytosolic form. So that's why the aminotransferases are important, not only just for making proteins or for getting rid of nitrogens, that we've already talked about, those two methods, but they're actually also important for using this shuttle of transporting electrons across the mitochondrial membrane. So it's an ingenious method to effectively get the electrons from the cytosol to the mitochondrial matrix. Okay. So that's why we've actually seen all this before, but hopefully it's starting to come together and light bulbs are starting to flicker. The little hamsters in your brains are going around in the little wheel. You're not like my brother that I call Scooter.
Scooter.
Yeah. And so, well, for a while we called him Tripod because his little hamster only had 3 legs going on its wheel, and now he's Scooter because as he gets older, he gets a little slower, and so we now think that his hamster just has 2 legs. If he loses another leg, he'll be called Roland. All right, so if you added up all the possible ATPs, NADHs, and FADH2s that were made, starting from glucose, going all the way through to, ATP, or I shouldn't say that, through the OxPhos pathway, if you use the glycerol phosphate shuttle, you get an effective amount of 30 ATPs from 1 glucose. If you use the malate aspartate, you get 32. But if you notice, there is no net gain or loss of either NADH or FADH2. The exact amount that you make is the exact amount that you get rid of. And so we have a very, very limited supply of these electron acceptors. Okay? And that's one of the reasons why we have to keep this homeostasis in control. And so you only have so much in this mitochondria, and you only have so much in the cytosol. You aren't making anything new because it gets used up, it gets consumed. Okay, then we have what are called the uncouplers, which these are actually very important. And what happens is the uncouplers uncouple the ETC from the OxPhos. So therefore, you can actually get electrons to be moved through the electron transport chain, but there's no net gain of ATP. That could be either good or bad. First of all, why would it be bad? That's usually the easier one to explain. Why is that bad? Right, you're going to run out of ATP. And so if you do this across the board, you're going to die. Okay, you're going to have no energy. When would it be good? Obviously, very limited cases. Pardon? Yeah, it's in thermogenesis because you're still generating heat. You're just not doing the work, if you want to think of it that way. It goes back to physics or Gen Chem 2 or whatever when you did some of the thermochemistry. Because those electrons are moving, so it's an electrical, function. So you can generate heat that way. And so that's what brown adipose tissue does. And so the way that this what I mean by the way that this works, I forget which example this one is. I think this is maybe blenomycin that they have there. But you can see what this is the colored parts are those that are charged, whereas the, silver, gray, or white parts are the nonpolar portions. But what does this look like if you're looking down it? Like a tube. I know I had one student say, it looks like you're looking down the barrel of a gun. It's like, please don't look down the barrel of a gun. But the inside is relatively hydrophilic. The outside is relatively hydrophobic. So what will happen if you get this molecule inside your mitochondrial inner membrane? It makes a channel. So essentially this is what it looks like. We've got, here we go, the membrane. I'm not going to draw all of the phospholipids. There we go. But then we have this channel, and it has an opening all the way through. And so you've got all of your protons, which are they charged by their very nature? Yes. And right here you'd have the normal ATP, synthase, which it's a turnstile. You'd have to do the work and go through the turnstile. And let's just think about it. Like, for example, whenever I was a student at Duke, Duke's football team now, in the last couple of years, has been good, and they made it to bowl games. Whenever I was there, they were atrocious. They went 2 years without winning a single game in conference. So they had the gate with the turnstiles, and then they had a door that was open. And so, what do you suppose the students did? Do they go through the turnstile, or do they go through the door? That's this is a big opening. The door, and that way you didn't have to pay. That, and technically they didn't charge you anyway because no one wanted to go to the football game. But and that's the same thing that happens here with the protons. You go through the easiest route; it just goes right on through. So you have the electron transport chain. Making the electrons. The electrons are going through there. They're pumping the protons out, but the protons are just going through the open door. They're not going to go through the turnstile and have to do the work. Why push the turnstile when you can just flow through the hole that's in the membrane? Okay, and so that's why you can generate the heat for thermogenesis, but you're not making any ATPs because it's going through that open door. Okay. And so there are some other examples, 2,4-dinitrophenol, phenylamycin, gramacydin A, and then there's the protein thermogenin, which is what's in brown adipose tissue. So the oxygen is still used to make water, but there's no ATP that's synthesized. So that's phenylamycin. You could see where, this one's really obvious where it's charged, and so the protons can just flow right on through. And so, these are antibiotics. There's also some toxins that, the reason why they are toxic is because they puncture holes in the membrane of the inner membrane of the mitochondria, and so therefore you don't make any ATP.
So what is the antibiotic?
Pardon?
Attack.
So then if you make an antibiotic that targets more of the bacteria, because bacteria, resemble that of mitochondria, then it perforates theirs, and so it circumvents their ability to make ATP.
So what is the dinitrophenol for?
That would be toxic.
Okay.
All right. So, ROS stands for reactive oxygen species, in case you didn't know. And that just naturally happens during OxPhos. We've already talked about heme B and how heme B is used to combat it with respect to complex 2. But we also have to have other ways, because it's just, like I said, when you're going 150,000 times a second, there are going to be times that electrons will fly off. Okay, and so this is just to show some of the possibilities here, right? And one of the major ways here is this superoxide dismutase, which there's actually multiple superoxide dismutases. Those are called SODs. And what they do is this, whoa, come on, it's frozen once again. There we go. This species here, this radical, this O2 minus radical, is called superoxide. And what superoxide dismutase does is it can convert it to hydrogen peroxide, which itself can generate radicals. However, it's much less radicalized than superoxide or the related hydroxyl radical right here. Okay, and so we have glutathione peroxidase, which is an enzyme in our body that is one that takes peroxide and converts it to water at the expense of glutathione. Remember, glutathione was one that we talked about in the past of using it for protein structures and helping make sure the protein structure is correct and not making incorrect disulfide bonds. But this is another way that it's important, is it also helps get rid of free radicals in your body. And then the glutathione gets regenerated by glutathione reductase. Okay. And so this is just to show you a little picture of what it looks like. If it's either going to be a free thiols or it's going to be disulfide. Okay. And so, in particular, superoxide dismutase is very important, and there are disease states that are associated with superoxide dismutase. Okay. Oh, and the reason why is the free radicals can damage essentially any biomolecule can be attacked. Okay. And so some of the mutations with superoxide dismutase are believed to be involved with aging. So just one of the reasons why people age. One of the ideas out there is that you have a generation of more and more reactive oxygen species. And then we have ALS, which what does ALS stand for? Right. Amyotrophic lateral sclerosis, and it's more commonly called what? Lou Gehrig's disease, which this is Lou Gehrig. Okay, and so, which actually I had a friend who died from ALS. It was one of my best friends in high school. It was her mother. And then there are also neural diseases associated with Down syndrome, because essentially everyone with Down syndrome will have the characteristics of Alzheimer's disease, and one of the beliefs is that they also have issues with their superoxidase mutases. And then there are certain cancers that are believed to be caused by a dysfunction either a dysfunction or a decrease in the catalytic efficiency in SOD. Okay, and so typically all of these would be fatal because eventually you accumulate enough of the ROSs that it's going to cause neurological damage. Are there any questions or comments?
How do you think, like, in the Bible that some people, like, lived a very long time?
So you want me to come up with a hypothesis on the fact that maybe they had, like, the Uber SOD, like, hashtag perfect caption idea?
I think it's because before the flood, no animal products were consumed. And people lived 700, 900 years. But after the flood, when it was, people lived.
Is it really true that no animal products were used?
It wasn't given until after the flood.
Right, I was going to say, they also talk about before the flood they ate lamb. That's an animal product.
There goes your hypothesis.
It's like, don't!
I would want to die if I was 969 years old.
Did, like, everyone?
Like, I want to die if I was
Well, who knows that maybe...
frailty or not.
Right, maybe he was having a really good quality of life for the first 968 years. Maybe they had a really, really catalytically efficient, superoxidase mutase. And what's another hypothesis with respect to aging? There's another enzyme, telomerase. So, because it's for the high fidelity for your chromosomes.
Wasn't,
I don't know.
Earlier, we were supposed to have, like, a higher concentration of O2 as well.
Quite possibly. I don't know. I never really gave it that much thought, honestly.
That was a good faith integration.
It was. Kudos. Kudos to you. You get the Metaphorical Cookie Award.